Okay. Thank you very much for the invitation. I'm going to talk about thermodynamics and transport across cell membrane. So again, it's without many equations, so it's more biological type of thing. Although there's going to be some Monte Carlo stuff. So basically, this is a scheme that we have in our life that we get natural resource, we build something, make some industrialization, production, then packing. This is how we survive in the city. And our body has about the same thing. Then we get the food, we do some reactions inside and produce heat. And basically, in this is a conservation of this the first law of thermodynamics. Then we have the conservation of energy. So whatever you eat, or you do work, or you produce heat, or you gain or lose mass. So if you eat more than we burn, basically we increase our mass. That's how we work. Basically, I'm going to talk a little bit about the foods of our that we use. I'm going to talk a little bit about work and have something about these heats. Let's start with food. So we eat everything. I mean, we eat carbs, energy, glucose, vitamins, fibers, everything. But I'm going to focus on the energy. That's more like thermodynamic thing. The macroscopic energy we eat, it's basically glucose, sucrose, or fats. Sorry, it's Portuguese. Most of these we can just calculate the energy inside using very simple relation of enthalpy of all the bonds of these connections that build the molecule. And then we have, for example, for glucose, we have the energy inside the molecule of glucose. If we burn, it's around 2,000, about 3,000 kilojoules per mole. This one, if we burn, mean that basically have a reaction of the entire molecule, reaction with O2, generating water and CO2. Basically, that's the energy we get. But inside our body, we use different type of energy because this one it's a lot of energy. If you see, I mean, covalent bond is around 600 kilojoules per mole. So if you burn this kind of energy, if you release this type of energy inside our body, basically we're going to break all, not all, but several connections, several covalent bonds. And this one is the ATP, which when you do some reactions here, when you neutralize ATP, then we have free around 28, 30 kilojoules per mole. So this one's more compatible. This one can work because if you see here we have in the ATP, we have around 30 kilojoules per mole. Covalent bound is around 200, 300, and the KBT is around 2.5. So this one can do some work because it's above KBT, but it's weak to break this type of connection, this type of bond. So that's most of microscopic energy we use in our body. I'm not going to enter this, of course, but in order to come from here to here, it's a long process. This energy, I'm telling you here, it's the energy if you burn at once. But our body have to break in this very slow process, which is the next slide. Okay. Oh yeah, it's getting slow. So from glucose to ATP, this is the Krebs cycle. Then it go inside the mitochondria and have a lot of process. I'm not going into this, but the fact is, for every mole of glucose, you have around 32 to 36 moles of ATP. So if you do a small calculation, if you get the energy of ATP and multiply for that amount, then we see that from 1 mole of glucose, we get around 1,200 kilojoules per mole of energy. And up here, we have 2,800. So then we see that only from this conversion from glucose to ATP, our body loses around 60% of the energy. It's a lot. So and this energy goes in form of heat. So basically, when we work out, when we do anything, basically we are doing this this process, which is burning, and burning. 60% is a lot. But if you go deeper inside, most of this energy, most of this heat comes from channels, membrane channels. So if you look, this is a very specific channel, like a pump, a CO2 pump, which is inside the endoplasmatic reticulum. So only this by itself, on a resting situation, one muscle cell can get around 40 or 50% of the entire energy of the muscle. 40 to 6% is burned out here. I mean, only in a single channel, only a single pump. So which means that most of this energy that we lose, a lot of our heat from our cells coming from this type of channel pumps and everything. So then in our group, we decided to go deep inside on this and study cell, but looking the food that will come, and the way it uses food for doing work and the heat produced. So basically, it's our idea. It's not finished yet. So we are going to making some steps towards this direction. So then what we have, let's talk about work. I told a little bit to you about food. Let's go a little bit about this work. So one of the ways to measure the work a cell can do, it's it's simple to explain, not that simple to do, but basically you have a cell here, and we mark the substrate the cell is in. And then instead to observe the cell, we observe the substrate. So the substrate is flexible, and we know the properties of the substrate. Then once the cell contracts, then it moves the beads that we are following, the fluorescent beads that are below the cell, and then we see the force it does. So basically, we can get this information, and I'm going to show a few animations here. So this one is one of the examples. Here's the cell. When you look at the cell by itself, these are cardiomyocytes, neonat cardiomyocyte, that's why they've shaped. And this one is the substrate after you do some calculation. It will work. So then you see it's contracting, and then it makes some force out here. So this one, but this one you can do for like minutes, and then you can just observe the force the cell is doing on the substrates. In other words, you can convert in work. And here is the cell itself. I mean, if you look, you see a little bit of contraction, visual contraction on the cell. So, and this is a graph. It's one of the output of this experiment, and we can integrate these results. And then we can, for example, compare how the cell behaves in a regular situation with nothing going on, and then if you put some drug that make the cell like adrenaline, and then the cell will beat faster. So these is two examples of the same type of cell when you do just a control and when you do some drug. So it multiply by a factor of 3 the amount of beats per minute. This one is one way to control the beat, the way it beat. This cell is a little older than the other one. So then we put one electric field between the cells. This one we built in our lab by ourselves. I'm not sure if you're going to listen to the noise. No, there is no sound. But if you could hear, we put like a sound like beep, beep, every time we have an electric field. And then you could see that this one is synchronized with the fields, but there is no sound, no problem. And this one is a branch of cell. This one is, each of these is a different cell. Now we can look also the behavior, the qualitative behavior. It's the same thing, but there's no sound. So, but then you can see like, this is almost like a muscle, heart muscle. And the interesting thing is it does the force more here and a little here. So these cells are working together to provide a huge force in the extremes. Okay, and about the food itself. So what we do is we can get the media the cell is in, and compare, because the media, we know that exactly everything that's inside the media we know, because we buy one bottle of this media with all the ingredients very precise. And then we put to the cell, work with it. Then afterwards we take out this media and go to a resonance NMR. And then on the NMR, we can identify everything that it was before and was after. So we can see what the cell, basically the ingredients the cell took from the media and what it adds on the media. So basically we can go backwards and see the cycle, the metabolic pathways the cell used. So we can get the amount of glucose it took from the media and basically how much work it does. That's our goal. And here, just one example of two spectra. This one is a normal cell. This one is a knockdown cell. This is a cell from actually, no, this one is the baseline, the normal cell, and this cell is one modified cell, a knockout cell. So this cardiomyocyte will generate a heart disease. So this is a sick cardiomyocytes. So then we can compare. This one is the metabolic we could collect, so we could see the difference, and then we could have some explanations of what is going on out here. And about the models, so since we know that most of the energy are from these channels, we decided to go a little bit deeper and go inside these channels and to see what is going on these channels and how can we model this and how can you do experiments on this. So I'm not going to talk about experiments on this. I'm going to talk just about the models to have some balance between biology and physics. This was a membrane, and the membrane is full of those transported, these channels or pumps that can transport some, let's say here it's a potassium, but can transfer lactose, glucose, everything from outside to inside, even across against the gradient of concentration. And there are several models. So these are simple channels. This has no energy involved. So it's just a channel that facilitates the diffusion. This one is an antiport type of seaport antiport of type of channel, which allow a high gradient of let's say protons or ion or any ion to cross. And because this one cross in favor of the gradients, it transport together something against the gradients. So, and this is a more complex type of motor. This is a ATP fast. I'm not going to talk about this, but basically, I'm going to focus a little bit out here. We could model, then we start to do several models. So we have three or four types of different types of channels and pumps that we model. I'm going to talk about these ones, about the lactose that can transfer lactose from outside of the cell to inside the structure, from outside to inside the cell. This is the protein database. From the protein database is the shape of the SIM-port transporter. And here is more or less how it works. So this is the lactose transporter. So it basically takes protons. There is a pump of protons to make this region with a high gradient of protons. And this is start to pump using ATP, it pump out. And then this gradient of protons goes down and bring together the molecule of lactose. That's how it works. And we start from one model like this. And the model like this involves that we get the configuration of all the states. So we have 6 states. We have all the configuration of them, and basically compare the energy that we could get from here to here, because from this state to this state, we know that only these groups of molecular of atoms will change position, and based on the change of position, we could have some clue about the energy involved from this transition from here to here. So from all these transitions, we could get more or less what is the step, the energetic step between one and the other one. And this is our initial guess. So we basically put this in our model as a guess, and then put the model to run and see if it works. And then we start to change a little bit these steps, these energies, until you see the system start to make the cycle, and this one generate the. So this is just to see. This is the initial energy of all of them, the six states, and then when we put the code to run, it starts to search, and then we find this. The final graph of the difference in energy between the states and the state of the lacing. I'm not talking about much equations. Basically, we run Monte Carlo simulation, and then we start to get the behavior. So this one is the fraction of lactose inside the cell, outside the cell, and then we see when we put the model to work, this is Monte Carlo step. That goes slowly and reach the equilibrium, and here's the same thing, it goes slowly to get the equilibrium. And this is the proton fraction, again, the same thing, it's outside, inside and outside, and you see that the concentration, the fraction goes up on this and down on this, because this one is facilitating the transport of the other one. And here it's a more long process, so we are just going to see how it goes in a very long time and following. And this one is each step of this, each circle of this is a full cycle. A full cycle means it goes from the 6 steps. But because it's a statistical model, it can go from one direction, and it also can go from the other direction. Sometimes it works in putting lactose from outside to inside, and sometimes there is a backwards. And in the dark one is the times the cycle went in the wrong, let's say, wrong direction. And the other one, the white one, is the correct direction. But you see on the average, it goes up. Meaning that the white one, it's getting more than the more time it goes from the correct cycle than the wrong cycle. And we also could compare these results with experimental data, because this curve we have more or less in the experimental data, and then we could find how much Monte Carlo steps correspond to 1 second. Basically, we could convert, based on this curve, we could convert Monte Carlo step to time, and then we get this type of curve. So now we have time against amount of lactose, and time against the lactose remaining on the periplasm. And the circles are experimental data, and here's our curve, which we made from our model. So our model fits perfectly the experimental data. As I mentioned, the model we did, the first model we did was like this, but then we realized that sometimes there is a shortcut. This from experimental data. We learned that there is this shortcut. Sometimes it goes that, and instead you drop the lactose outside it back and drop inside. And then we add in the model this type of transition. Then you start to have results. I'm not going to show all the results because we wouldn't have time. And just to show the 4 types of models we are working on. This one is another model that we just add. Instead to have this connection here, we put other one here. And this one is for a completely different type of protein. So basically, we built this way to reconstruct, not reconstruct, map. Actually, I would say map. This is my last slide, so it's I can just talk, and then when it reach my time, just let me know. What we did is, in terms of modeling, we have one way to map basically any type of those channels in a Monte Carlo way. Then we are testing how can you do with different ones and how it fits, how it goes, and try to basically understand this question, which is what about the heat? So what about the heat is our next question, because basically we could see the amount of energy the cells are getting from the media. We are able to see the amount of work it can do. Although we have still to generalize, we are getting for a single cell. So then the idea now is to have one way to, from not one single, but from like 10 or 20 different cells, we generalize and get one approximation of what is going on in the entire system. And now we are interested in measure this heat that is going out and compare what expected from the model, because from the model, we also know the energy that is in the gradient of concentration between the two. So we more or less have some idea the amount of energy that's not going to it's not entering the process. So this energy must be going out. So that's our next step, is to include heat on the process. Is it?
Adriano, maybe I have missed it, but I was in doubt about your Monte Carlo model. Is it a minimal or a no whole atom model?
What do you mean minimal?
Lattice or
No, actually, see.
grained, or what is your?
Let me just talk a little bit in this. So we don't have like a lattice type of model. It's basically we have what we call this one chamber, and this chamber has one number of let's say n particles of sodium, n particles of lactose in this chamber, and this other chamber the same thing, and all stages are other chambers. And then we basically look what is the probability of one element in the side up, for example, be close enough to the channel to be captured to this. And it will be proportional to the concentration. Then after that, if let's say the lactose is captured, then we see what's the probability then a proton also be captured, which would be again proportional to the concentration. And once they are there, we see what is the probability to move from this one based on the free energy difference between the two states. And then go. It's in using the regular Monte Carlo, but it's not that it's the particle has a spatial location. The particles are inside one chamber and like a mean field, like a, I should presume say mean field type of Monte Carlo. It'll be easier. Sorry for the long way to say the correct sentence.
Showed all atom membranes. So.
Yeah, no, that one is. We also are doing some calculation using molecular dynamics. We are doing molecular dynamics, but it's not ready to show. I mean, we have some results, but not ready to show.
You alluded to it at the end of your talk a little. I was curious if there's any cooperative effects among cells. And I guess if your models mean field, probably not. But you know, like I'm thinking if like one cell is more efficient or there's some heterogeneity in cells. So if I couple them together, yeah.
Yeah, the heterogeneity is a huge heterogeneity. And one of the main points of this the heterogeneity is that the cells sometimes it can have a different size, different. Sometimes we put these fields, electric fields, and one of them just like do, others don't. When you have all of them together, then they are completely collective. When you have that structure that is thousands of carbon monoxides together, then they beat together. And then there is an internal electrical potential action that goes through them. Then it's
yeah, just to follow up, the question is whether kind of a model like the one that you just showed, if you coupled a bunch of cells together, something like that would emerge. Like in other words, sort of like when you had a large n limit of that system, if there was some coupling between them, if you might get some interesting sort of, you know, collective emergent behavior.
Yeah, probably, but we didn't go through. Although, I mean, we are doing now in terms of experimentally, we are doing another step, which is even, well, this will be something between, but somehow we do stuff, which is mixing different type of cells, because basically in our heart, in that part of our body, it's not a single cell. We have like a cardomyocyte, a marcapasso. I have no idea how to say.
Pacemaker.
Pacemaker. Oh yes. Pacemaker. They are together there. And if you go to different parts of our body, again, I mean, the muscle has other cells together. So you're going to mix to see the interaction between them. But from an experimental point of view, from the point of view of model, we are really starting from the bottom, which is molecular dynamics, then single channel, to try to understand this and trying to go to put different channels together. Right now, we keep the concentration of protons fixed in one side sometimes, or fixed in the sense that there is no pump putting back. It's not fixed because it goes through, but then we don't have the pump to go out. So our next step is try to make a complete system, because once you put the pump, we know the amount of energy the pump uses. Then we can see the energy. That's our next step. It's putting the pump and see the flux of energy and try to understand the amount of heat it's supposed to get out, and try to measure it with the challenging thing.
Yes, thanks for the talk. I was just curious, so in the first part of your talk, Egypt were measuring the intake of glucose, right?
Mm-hmm.
Or not?
I mean,
The first part of the talk, so you said you added the and measure the intake of glucose, actually.
Yeah, from the cell, the amount of the amount of glucose the cell
That was glucose.
yeah, glucose.
Yeah, so why did it, in the second part, why did you put the
Lactose.
glucose transport instead of lactose?
Yeah.
Glucose has glucose, but I mean, there are glucose
I
transport.
mean, see, science is not the way it's supposed to be sometimes. The point is, the lactose type of transporter, it's like way before we did the experimental part of, so the lactose part starts from master degree students. Now, like 6 years old, we start with this, then we start to move. And then more recently, then we start to take this experimental part where we did an MR to make the measurement. So this more recent. And then what we did is to move now the experimental model to go closer to what we're getting, like the basically the channels which use more energy for this.
So that the structure of the channel matters in your Monte Carlo simulation.
The structure of the channel, yes, because every step, yeah, every step. So first of all, glucose and lactose, they are very similar. The transporters are similar. It's not much difference between them. Some difference, but not much.
Right.
But what we did, it depends on the structure because we compare to get the free energy difference between each state. We compare the structure, the BED of the two structures,
Right.
see those connections that's in one and not in the other one, and looking at the energetic part and get the energy between the two of them. So it depends on the structure to get this. So in order to do the glucose, we have just to redraw the first map and try to get the same thing. And I guess it would be that difference. But the reason they are lactose and not glucose is historical, because one starts first and the other one. And the glucose is what we get on the NMR.
Well, maybe you can just use a medium of lactose. So.
But then the cell must be different. I mean, it's not the type of, I mean, I think it's easier to change the model to glucose than the opposite way. I think it's a more.
I have a kind of naive question or vague, I don't know. You have some results that are very relevant for your biological system, and you are using this model that's interesting. And I have seen that I was mentioning to Andrea, for instance, that you can study this kind of models using non-equilibrium thermodynamics, for instance, like Carlos Fiore.
Yes.
And something like that. My question is whether you can find something interesting from the point of view of non-equilibrium thermodynamics from these models. There are elements there.
Yeah, actually,
Yeah.
I talked with Fiore that we should do something together, try to understand this. It's not I'm not doing a lot of non-equilibrium simulations. It should be very interesting. But again, I mean, those things I'm connecting, as I mentioned to Antonio, I'm connecting now. I mean, because a lot of those works we're doing with different parts of my group. I mean, some students doing one thing, other students another thing, another student another thing. And then once I think last year I decided, okay, let's try to see how can we converge what everybody's doing in a single stuff. And then what I'm showing here is what like the group, I would love the group would be. It's not the way the group is right now. I mean, it's two things. I mean, if the group, this wouldn't be the perfect situation. I mean, one group of students doing this thing that is connected to the other thing that's connected, but was not the way everything went through. But that's good. I'm going to show this one in the lab. And you see people, you have to connect this because it's missing some small. But we are submitting a project FAPESP to
look into these other contexts, right?
Exactly, and introduce different type of models and everything. I mean,
So Andrea has a question. Back on the actual report that you was asking, we have that imbalance between influx and efflux. It looks like there's some kind of fluctuation field.
Yeah. Yeah, yeah.
Maybe connects with the.
Yeah, I mean, it's again, I mean, once you do the probabilistic thing, it goes and sometimes it backwards. Yeah, it's a lot of fluctuations here. Yeah, and
Probably you mean that
Yeah.
you already have data.
It's more of the imbalance could be some kind of consequence.
oh, you mean that I already may have the data
To analyze the fluctuations and look at some, perhaps you mean some thermodynamic uncertainty relations or stuff like that.
No, more in the case of the HRC's fluctuation theory.
Okay.
Connect that with the entropy production.
Okay.
Yeah.
So it would be kind of a consequence per second.
Yeah.
Yeah. Yeah, I mean, yeah,
It could be a connection.
yeah, I could look at it. I mean, it's interesting, yeah.
In the case with many cells, you have a synchronization of
Yeah.
activity.
Yeah, it does.
Perhaps a model of synchronization such as Kuramoto's model.
See,
oscillators
I mean,
could be-
let me talk a little bit about the synchronization out here. Those cells that I was looking was a neonate cardomyocytes, which means that they have the peacemaker inside. So those cells, you still didn't decide which of them will be muscle itself and which will be so they still are going to differentiate later on. So let's see, most of them has some properties like a peacemaker, properties like a muscle. So they contract and they stimulate. In the real heart, they are completely differentiated. Then we have the pacemakers and the muscles. And they do the synchronization. So they talk very fast between them using channels like a sodium, which is very fast. And then they communicate with the muscle with calcium channels, which is slower. So they have an internal communication between the pacemakers and the muscle. So the synchronization has one kind of a neural network. It's not neural, nervous type of network that will work together and do this. And sometimes, for example, one of those peacemakers can be crazy, and then you have arrhythmia. And then you go to a doctor, go there and burn that peacemaker to kill them to avoid this asynchrony.
So what you're saying is that not all of them have the same natural frequencies
No.
as oscillators in the experiment.
Exactly.
But you still can see synchronization.
Because you have one that is doing this stuff, doing this. So those cells can beat even without any stimuli. If you just put there, they will do this. But they will do like a click and then wait a little bit more. So and then we put the electric field just to synchronize and to have all of them doing the same work. It's like if you go to do a exam, a test, a cardio test on the medical in a clinic. So they are not going to ask you to run on Ibirapuera. They are going to ask you to run in a fixed place. Then you're going to run there and treadmill, and you're going to run there, and they will put the velocity you should run until you can't run anymore. So they will change the velocity. That's what we're doing. I mean, we do these fields because we want to force them to have the same type of protocol in our experiments rather than they decided what to do, because then they're going to add one biological variability that it's not worth to include in our experiments.
Okay, let's take this. Sandwiches are downstairs already. I can solve that. I think so. Or we'll put all this in practice, right?
Yeah.